It's my pleasure to be invited. Thank you very much, and thank you very much for the wonderful introduction. Um, so I didn't realize bioactivity has been here for 15 years. I guess time flies, right? So I remember, I attend bioactivity quite regularly. Well, if I go to the KDD conference. So today I will talk a little bit about deep learning. We'll talk about the application of deep learning in cheminformatics in drug discovery. And then I will share with you our own experience about use deep learning to predict bioactivities of chemicals. And then I show you a web portal we developed for people to use. So probably people see this type of figures all the time. So the message you're trying to say is that neural network has been there for a long time. Of course, it somehow lost the momentum. So you can see the red line is the popularity of the term using the Google trend. And the big data and the deep learning are definitely picking up. So, so what's new? And let's look at the typical neural network architecture. I don't know why the image quality is, the display is not that good, but so bear with me. So you can see that here, first it's a multi-layer neural network, right? So you have the input layer, then you have the multiple hidden layer, finally you have the output layer. Each layer is a set of units called neurons. Each neuron is basically nonlinear transformation, and you can have many ways to transform the input data, right? ReLU or sigmod or other different type of nonlinear transformations. So the training part of the neural network, the difference between neural network usually is a few limited number of layers, and now deep learning trying to add much larger number of layers, and that turns out to be a very significant improvement. We are going to just briefly talk about that. So training neural network is just to optimize the weights for desired objective function. And then prediction, once you train all the weights, you can feed your data and then make predictions. So why we talk about neural network? Well, it trains sequentially. It saves you time. And also, it's this intuitive connection to biological systems where people believe there's some connection to our brain. Okay, maybe the connection is shallow, but we don't know. And it handles nonlinear relationship very naturally. Of course, now we're going to feature one specific perspective, which is they're trying to learn new representation of the data. So we view neural network as a way to generate features that not necessarily embedded in the original data, but that makes a lot of sense for the regression or the classification that you're trying to do. So before we talk about that, let me give you a very simple example. So here we have a 2D case. Here we have a 2D plane. So we have two features, X and Y. And I showed you red versus yellow. And of course, the decision boundary in this case is very clear decision boundary. There's no uncertainty. You have the line separated red and yellow. So if say we use decision tree trained on this model using X and Y and input data, what type of decision tree are we going to get? We're going to get a proxy, because using decision tree, you will discretize the X and the Y. So you're going to approximate the linear decision boundary by those blocks. So you never achieve perfect separation. Okay. However, if we be able to learn a new representation, just learn a new feature X plus Y, then you can separate red and yellow very easily. So this is a very simple example to show that the capacity of decision tree without feature learning. And if you do have a feature learning capability, you can do the classification very easily. So what does this mean for neural network? Let's look at another example, very simple example again, image annotation. We're trying to recognize the character 9 out of this image. So if we use a neural network, what we're going to do, each neuron basically is a feature detector. So let's look at this one neuron. Suppose it pick up signals from the first few cells. Okay. In this case, basically the neuron trying to recognize the horizontal line at the top of the image. So imagine that you may have other neurons which can pick up the middle horizontal line or the vertical line. And when you put all these signals together, when you build another hidden layer on top of those the low-level hidden layers, you may have something saying, if you have two horizontal lines and one vertical short vertical lines and one long vertical lines, you may get a digit of 9. Okay. You may also get a digit of 6. Then you need to say the vertical line needs to be, the long vertical line needs to be on the right to the left to the short vertical line. So you can see that if we build those, be able to detect those high-level features, then we put all those features together, we can recognize the characters quite easily. So deep learning basically trying to learn multiple layers of in an abstract way to represent the original data. That's one way to look at deep learning. So now we're going to, let's just give a very quick overview of what is deep learning. Now we're going to shift the gear a little bit to study the application of deep learning in bioinformatics. And specifically in this case is drug discovery and cheminformatics. So any questions so far? Okay. One thing about deep learning is that the great power means great responsibility. If you use a nonlinear transformation to generate additional features from the original data, you may have a very powerful capability to separate those cases not easy to be separated. However, as people are familiar with the machine learning and data mining, especially the supervised learning part, is that overfitting becomes a very serious problem. So if you be able to build a very sophisticated decision line, then you need to worry about that, whether you run into an artificial effect or some true signals in the data. And also the computational computational time, and also one thing about deep learning, a lot of hyperparameters to optimize. So I'm going to talk things a little bit about the regularization part, the control overfitting, and then we'll talk about how do we deal with hyperparameters. So one way in general to deal with to control the complexity to avoid overfitting, the L2 regularization. So you basically control the weight that you're going to assign to neurons, or alternatively you can do dropout. So what is the L2 regularization? So here, for example, if we say we want to do regression, and we decided to use the least square fitting as an objective function. So you have the predicted value, you have the true value. You look at the difference, you square it, and then you do a summation. So in order to avoid overfitting, what you can do is that add another component to all the L2 norm of the weighting vectors. In this case, that you're basically saying, you have to have a trade-off between the loss function and the complexity of your model. And alternatively, another very, very popular way to do regularization is dropout. So you randomly select a subset of neurons. You just make sure they won't produce anything output. And then you can do this. It's very similar to the idea of random forest. You random select subset of features. Here, you random select subset of neurons. You train your model, and you build an ensemble on top of this individual build models. So deep learning, they have, other than the multi-layer neural network, there are other type of architectures. We won't go to the details of those architectures. If you're interested, you probably can Google search. And now convolutional neural network start to find applications in bioinformatics and chemoinformatics. So there are a few. Here I make a list of commonly available softwares you can use. For example, Café from the Berkeley lab. They are original developed for computer vision architectures, but also they allow you to engineer or experiments on different architectures, and they all support that. There are Theano, there are other different type of packages you may use. So what are the challenges? How if we apply deep learning to bioinformatics, in this case, chemoinformatics, what are the main challenges? One thing we identified is that although there are many open source software, but they are very difficult to use. So, for example, the Café from the Berkeley Vision Lab, they developed this package with the computer vision application in their mind. And then when you the bioinformatics application, for example, protein structure data, crystallography data, DNA sequence data, and chemical structure data, they are not necessarily adopt the same format. So you have to, be able to hack into the code, taking care of the data format part. Secondly, that there are many hyperparameters, we are going to talk about that. Then how do we have a systematic way to optimize those hyperparameters and then build the best model? So now I'm going to share our recent experience about applying deep learning to chemoinformatics, and we are going to talk about application drug discovery and drug development. So before I talk about the deep learning, let's just quickly overview what is drug discovery and what is drug development, and what are the data analytics challenges in drug discovery and drug development. So here I showed you a pipeline of typical drug discovery and drug development pipeline. So here the first block is the preclinical phase. What you do is that you do basic biology. You identify either something called phenotypical screening. So you say, okay, I'm going to concentrate on breast cancer. Pick up the breast cancer cells. I'm going to develop an automated screening pipeline so that I can quickly test, between. Here I use a very small number, 5. Typically, in pharmaceutical company, test between 1 million to 3 million compounds for one particular disease. Or you pick up a protein, say, after 20 years of biological research, we know that this protein, if you can modulate the biological activity of the protein, you're going to have a cure of the disease. Okay, so then you start with 1 to 3 million compounds. You go through something called high throughput screening. You test the activity of this compound one by one. Then at the end of the day, you compile a very long array using the compound and its activity. And based on the reading, you decide to say, I'm going to choose 5,000 or 10,000 to go through my confirmatory screening. So then in this case, you purify the compound. You test them again, but not on a single concentration. You test them at different concentrations, usually 7 to 13 different concentrations. You look at the response of the chemicals, and you decide something called IC50, so the concentration that shows 50% of the activity. And then, based on that, you started to try to look at the chemotypes. You're trying to optimize the structure so that hopefully the toxicity or off-target effect of the chemical is minimized. So after that, finally you come back to about 3 to 5 chemicals, and at that point you are ready to do your clinical trial. You write a report to FDA. You say, this is how I'm going to recruit patients, and this is how I'm going to do clinical trial, and this is the method we are going to use to analyze the data. So you go through clinical trial phase 1, phase 2, and phase 3, and then you have this truckload document shipped to FDA. It takes FDA usually half a year to 2 years to review the document. And the result, if the FDA approves the drug, well, boo, the stock market of the company increases. Everybody open the champagne and celebrate. And also it's great news for patients because it's a new drug come to the market. And on average, how many drugs will come to the market? Probably, about handful each year. So it's not, those things doesn't happen very frequent. Why? There are many reasons. So I'm going to actually briefly touch that. So at the beginning of the drug discovery is high throughput screening. You're trying to test as many chemicals as possible, as fast as possible, to test whether they, one is whether they bind to the protein specifically, two, or alternative, whether they produce a phenotype that you want. So, and you can divide the things we talked about before, into something target identification, target validation, screening, development. We won't go through the details. So and each and every step, actually informatics play a role. Why? Because overall, it's a very risky process. It takes a lot of resources to develop a drug and try to bring to the clinical phase. So here this is a breakdown of the cost. So for preclinical, usually the cost about $400 million, and the clinical trial may cost another $500 to $700 million, and the total cost for a drug is between $500 million to $1.5 billion in order to, that's the R&D cost. And you can also break down, depends on what type of disease you're trying to cure. Okay, so what's going on? Is that drug discovery and drug development process is a very resource-taking, time-consuming, and therefore informatics actually many informatics approaches are developed to try to help out the whole process. So here I list a few recent papers using deep learning trying to help drug discovery and drug development. So for example, there are people trying to look at the solubility of the chemicals. They're also trying to look at the side effects, trying to look at the drug-induced liver injury. That's one of the major concerns for any drug development, to see whether they have a damage to liver. And there are other types. So in this talk, I want to feature two recent competition, international competition, one organized by the by a pharmaceutical company called Merck, another one organized by NIH in collaboration with EPA and FDA. So the first one is the Merck, is called a molecular activity challenge. They're trying to predict the binding between chemicals and proteins, and the EPA trying to predict the toxicity of chemicals. So although drug discovery and the drug development is different at the conceptual level, it's quite different from environmental protection. But however, toxicity is a key component connecting all the things, because from pharmaceutical company perspective, a drug is a chemical that show efficacy towards the disease, but show low toxicity. And if you can very easily you can get a chemical that will kill cancer cells, but at the same time they'll kill normal cells. So rather than get a drug, you get a toxin. Okay, so the reason I want to feature both competitions, that it turns out that deep learning works very well. Scored, actually, they got the top 1 position in both competitions. So this, I think, a very nice case to demonstrate the capacity of deep learning. So let's look at the Merck Molecular Activity Challenge that did a couple of years ago. So they are different. Here, it's very it's very difficult to read. I understand, I recognize that from screen, and if I'm going to share the slides and also their references, so you can get, if you're really interested in the particular protein or particular pathway, you can get information from there. So they are target. So this is a target based. So the target based means the particular protein, and they're trying to predict whether the chemical bind to the protein or not. And the ADME related to don't ask me what is the ADME. So relate to distribution, relate to metabolism, relate to the basically how the human body metabolize chemicals. So if a chemical stay in human body, always accumulate in human body, that cannot be a drug because it may have a very effective way, and it may have the low toxicity at the beginning. But if the chemical never get excluded from human body, eventually it becomes toxic. So there are many. Each line here is an assay. In an assay, you basically screen, you test a number of chemicals. You see which one show the desired effect. So here, for example, for the first assay, it's a metabolism assay, and they test 50,000 chemicals. And then they look at the positive ones, negative ones, and then they release the data, ask whoever built a model to use that data to train a model. And eventually, they release the testing data to make a prediction. So this is a result published by, I think, the Toronto team about their experience. And they show that deep neural network is consistent better than random forest. Actually, I think they get the #1 position in that Merck competition. It's a quite, if you go through the list, you will see that actually, if the number get a bold font, that means it's better than random forest. It's actually almost consistent better than random forest, which is very impressive. Random forest is a quite a generic method and has been applied to many different places. Okay, so 2 years later, the EPA and the NIH and the FDA worked together, organized this Tox21 data challenge. So basically the idea is that we're trying to use crowdsourcing to collect the data and then release the data to the research community. You can use whatever method to build whatever model you want. Then the organization committee release the testing data, and everybody make a blind prediction, and then the group tells you who have the most accurate classification. Okay, so what's going on is that they trying to predict toxicity of chemicals. EPA has been doing this. EPA has been doing this for a long time. So people for working on graph mining probably remember a graph mining dataset released by EPA to again try to predict the toxicity of chemicals. It turns out it's very difficult. If you just look at the structure of the chemicals, you're trying to decide the toxicity of chemicals, it turns out to be very difficult. So what they did is that in this case, the EPA pick up 2 biological pathways. For each biological pathway, they pick up a number of proteins, and they pick up a number of targets. So the idea is that they're trying to ask you to do 2 levels of prediction. One is that for per individual protein, you're trying to predict the interactions between protein and a chemical. Second is that you're trying to predict whether that chemical modulates that biological pathway. So the idea is that from the EPA's perspective, is something called not only structured data but also in vivo data to try to that turns out to better prediction of chemical toxicity. So basically they have 12 endpoints. Those are 12 endpoints are individual proteins, And then they have 2 biological pathway assays. Okay. So this is the result, again, it's not from our paper. We also participated the same competition. It's from the, I think, again, the Toronto team. No, this is a different team, but they use deep learning. So the Merck competition from the Toronto team, they use deep learning. And this one is from different universities. They also use deep learning. And all the paper published in a special issue called Frontier in Environmental Science. So the EPA group solicited the top performers and asked them to write their experience and publish that journey. So here, the top method, although they say that our method, that's basically our method to that paper. It's not my method. Then here there are 12 endpoints. And then in addition, there are 2 biological pathways. So deep learning turns out to works very well on a number of assays. And we also participated in the competition. The Frozen Arm and the ToxFit, the 2 groups from, one is led by a postdoc with me, another one led by a graduate student with me. And we use random forest and the support vector machine and Naïve Bayes. So our model is ranked in the top 10, but and also close, but there's still some difference. So so now at this moment, I just share with you existing stories of deep learning in cheminformatics. Then I'm going to share with you our own experience about applying deep learning to cheminformatics. Before I do that, any questions? Yeah, to make sure I still have time. It turns out there are a lot of, you don't have to rely on an international competition to get your data. Cheminformatics or drug discovery is a place now, traditionally, there's no way you can get real screening data because all the screening data are owned by pharmaceutical companies and they never release the screening data. When it comes to clinical trial by FDA, by US law, you have to release all the data, but you won't release the screening data. However, NIH recently established a consortium that is called MLPCN. If you're interested in that, feel free to talk to me afterwards. And we participated in MLPCN. Basically, NIH established national capacity for the research community to do high throughput screening. You cannot screen 1 million compounds. Usually, NIH's configuration is 300,000 chemicals. However, the good news is that all the data are publicly available. So you do have now access to high quality primary, primary screening cannot be high quality by definition, but at least the real data. And you can access to high quality confirmatory screening and secondary screening. So that's a good news. You actually have access to those data. So what we investigate is that we ask 2 questions. One is that if we optimize deep learning and also other machine learning algorithms like a random forest support vector machine, Naïve Bayes, or even the graph kernels, for people working on graph kernels, you know that cheminformatics is one of the driving applications for graph kernels. So there are many different ways to design graph kernel. If we optimize all these machine learning algorithms, do we see benefit of deep learning? Do we see that deep learning outperform other methods across a wide range of endpoints? That's one question. Second question is how do we optimize the deep learning hyperparameters? As I mentioned, there are many parameters. Which activation function do you use? Which regularization do you use? How do you set up the regularization parameters? How many layers are you going to use, and how many neurons you are going to use? So that's 2 questions that are somehow related. So what we did is that we collect a large number of bioactivity data from different resources, and we systematically divided them into training and testing data. In the training, then we use the training data trying to optimize all the parameters using, again, cross-validation on the training data. Then we build the models from the training data. Then we apply the model on the training data to the testing data that we never use the testing data as part of the training. So you apply to brand new data. and record the performance. And we do this systematically to both deep learning and to other methods. And we're trying to see what's the effect. So here I list the data set we have. They are from ChEMBL, and they are different. They are target-based screening, so they are different proteins. And then the one thing about ChEMBL is that usually the quality of data is high. So if they say active, usually it means positive. It's positive. And one thing about the chemical informatics is really difficult to get negative data. So once you finish a screening, you don't see activity. People stop that at that point. They won't test it a systematic way, using a lot of resources to confirm it's not active. So what we did is, that's a problem. I think nobody has a good solution. So what we did is that we pick up decoys, we pick up random selected chemicals that with a low chance to really interact with that protein. And here, so there are 7 data sets, and they ranging from a couple of hundred to a couple of thousand confirmed positives. Then we generate synthetic negatives. And we controlled the molecular weight, we controlled the IC50, we did a lot of control to make sure the quality of data is high. And then we compared, so in order to have a fair comparison, we want to optimize the performance of deep learning. We also want to optimize performance for other machine learning algorithms. So I make a list. For each method, I make a list of hyperparameters. For example, random forest, number of trees, and how do you build the decision tree? You're using gene index or using entropy, and then maximum features. For supported vector machine, which kernel do you use? What's the parameter C? This magical parameter C, and then there's a gamma. And for neural nets, we also list a number of parameters. So here, there are a few critical ones. One is number of layers. When you build a deep learning, how many layers are going to use? And then for each layer, how many neurons you're going to use? What's the activation function? So we tried ReLU, sigmoid, other things. And then do you have to use regularization? If you do use regularization, how? And then what's the parameter? So typically, this is give you a summarization. We don't use a grid search. That takes too much time. If you list all the parameters, you list all the possible values. If you do grid search, combinatorics just kills you. So we hand pick up 48 configurations we think are reasonable and cover the configuration space. And then for random forest, we pick up 24. For supported vector machine, we take 20 configurations, and Naïve Bayes, we pick up 6. So what do we mean by this 20 configurations for supported vector machine? For example, we can pick up a combination for the C and the gamma. Okay, so each configuration is one particular setting for the hyperparameters. And then so we have 7 data sets. We did a cross-validation on the training. So we first divided the data set into training and testing. And in the training, we use cross-validation to evaluate the performance. And then finally, we build on the testing data. So we run about overall about close to 30,000 jobs in order to do the performance evaluation. So this is a quick result. First, we compared the activation function. So here it turns basically ReLU turns out to be always better. So so here there are 3 different activation functions. ReLU, sigmoid, and the reverse function of tangent. And then we have 7 data sets. So what you see is that 7 blocks. Each block has 3 boxes. It turns out from these results, it's quite consistent that ReLU performs better than the rest of the activation function. we use the MCC as a way to evaluate the results. The reason is that for the testing data is usually unbiased. It's usually very biased. So basically the ratio between the positive and negative is always there are always a small number of positives compared to the negative data set. So then the second question is that how do we I think let's say. So what's the number of layers, hidden layers we are going to have? We first see what if we use just a single layer, and then we use two layers without regularization, 2 layers with regularization, and 3 layers with regularization. It turns out that so each line here is a data set. We have 7 data sets, so we have 7 layers. We have 7 lines. And then we have, for each line, we have 4 points. It turns out when you move from 1 layer to 2 layers, most of the time you see performance gain. And when you move from 2 layers without regularization to 2 layers with regularization, you always see performance gain. But when you move from 2 layers with regularization to 3 layers with regularization, you didn't. At least we didn't see much performance gain. So at this moment, we see, based on the data set, we see that there's no benefit if you move beyond 3 layers. And here we also showed for, again, it's very difficult to read. I'm happy to share the slides. So for each layer, how many neurons so we test 50, 100, 500, 1,000, 1,500 to 2,000. It turns out that basically the conclusion that a couple of thousand, between 1,000 and 2,000, you see the maximum benefit. If you further increase the number of neurons, you slow down your training phase, you slow down your testing phase. You don't see much performance gain. So in addition, we run the experiment is that how much data you use for training. We did it at 5% and 95% split. So we use 5% of the data for training, 95% data for testing. Then we shifted that actually from 1%, then 5%, 10%, 30%, 50%, 70%, 90%. With the more data you allocated for training, the performance you build better models, right? The more data, the more training data you have, the higher quality models you will get. And it turns out that about at the value of 30%, we started to see the performance gain start to saturate. So when you have about 30% of the data, so first from 1% to 5%, you see a significant performance gain. And from the 5% to 30%, you also see quite a significant performance gain. After that, you don't see much performance gain. So that's a very interesting phenomenon. I think one of the advantage of deep learning is that it works with a large number of data. The more data you have, the better model you potentially can build. And I think that's the reason the deep learning become very, very popular. That's one of the source of the power of deep learning. However, for these data sets, for the one we have, we don't observe that. We don't see a consistent moving up figure. It's still saturated at a certain point. So that I think that's something we're planning to further investigate to see why we didn't benefit from additional data points. So here we showed you how do we optimize the deep learning part. So it turns out 2 layers with regularization using ReLU as the activation function, you always introduce regularization. That seems a reasonable across-board configuration that works with a large number of data sets. Then we use the same procedure to optimize to build an optimized model for random forest, supported vector machine, and Naïve Bayes. Then we compare the optimized performance only to see what's the benefit of deep learning. It turns out across data sets, So here we also showed the p-value. So it turns out that deep learning kind of like outperform all the methods inconsistently in all the data sets, but the performance gain is limited. We don't observe a huge performance gain. However, the p-value is significant. So although the difference may not be huge, I think that observation is consistent with other published results. So deep learning outperform other competing methods, but the performance gain is limited. However, it's statistically significant. So let's do a quick wrap up at this moment. So ReLU is the best activation function. 2 layers seems sufficient. Dropout regularization definitely helps. And it turns out 1,000 to 2,000 neurons is sufficient to build the models. There are few ongoing activities. For example, I mentioned, with the increased number of training samples, we don't see the consistently increased performance. So we are trying to investigate why. Secondly, that for chemoinformatics data, the quality, no matter which source you obtain the data, there are always this concern about the quality. So we're trying to see whether the deep learning is very sensitive to the outliers or the mistakes in the data, or it's not that sensitive. So I think I'm probably close to the end. So what I'm going to, I don't have internet access here, but what I'm going to show you is that a web portal we developed for the using deep learning to chemoinformatics applications. We call it the Chemo Cafe. So the Chemo Cafe, the URL is here. You go to my website, you will see I will put the link at my website. So the interface the interface is very simple. First you have to register yourself. All the registration will ask an email address. And once you register yourself, you can upload your data set. The data set is very simple. For chemical structure is something called a SMILE string. It's a string representation of the graph structure of the chemicals. So you get your SMILE string that tells me the structure of the chemical. You separate it by comma. You use either 1 or 0 to indicate it's active or inactive. And you upload the data set. And then we're going to, let's see. I don't think yeah. So if you upload the data set, we provide 3 configurations you can run. You can change, Yes. Yeah, I'll try the KDD. Is that KDD 2016 ban? What is the ban?
The exclamation.
Oh, okay. So I didn't see that.
Yeah, it was incorrectly invoked.
Yes, exactly. Okay. So you can upload. Click here so we provide sample files. If you try, you don't have to have chemical structures. So we provide sample files. I already downloaded that. And we provide an interface. You can change the dropout, or you can change the hidden layer dropout. You also can change, say, I want, here I build a model with 2 layers. I'm going not, I also can compare a model with 3 layers. And for each layer, how many neurons you include. Then you can upload a separate testing file. you will say, I'm not a robot. This is the case that straight numbers. I think I just clicked the wrong one. So click the new images. Select all images with straight numbers. Okay, so it seems it's difficult to pass the whoops, I think it's too sensitive to the houses. Okay. So yeah, I'm sure I'm sure I'm a human being. Are you sure you talk to a human being at this moment? So also click on the new images. Okay. What? This one? 3 may not be a. Finally. Okay. So here we allow you to select. You can, if you don't want to build a dropout, you can put 0, or you can put some certain value of dropout. So we allow you to select 3 configurations, build your model. And if you click submit, what you're going to do is that send the data to our server. We're going to build the 3 models and then give you the prediction. So for the training data, we'll give you the statistics showing the F1 score, MCC, AUC. And then for the uploaded validation data set, we'll give you prediction results. So I think that's the end of my talk. And I'm ready to take questions. Yes?
So you mentioned that you use a 2-layer hidden layer.
Yeah.
Learning. Have you thought about increasing the number of layers? Because
What?
that's like have 8 layers, and by increasing the number of layers, suddenly you have much more power to them.
Right. We tried 3, but we don't see benefit, and we stop at that point. So yes, we're trying to now increase to further numbers. Yes.
What was surprising with the Merck challenge was that they framed it as a transfer learning problem, and they reused their main network across targets, and they just retrained that bottom layer for each individual target. That was kind of surprising to me because binding sites are very different across targets, right? So
Sure.
one compound being active in one target may not be true in another target. So have you explored exploring this as a transfer learning problem, or what are some of your thoughts on that?
We did transfer learning separately in other applications, but we never tried in this setup. In general, as you mentioned, it's quite a challenging problem. There are about at least 200 GPCRs, right? And each one has its own binding profiles. So in order to transfer knowledge from one GPCR to another GPCR, it's difficult. Set along from GPCR to kinase, that's almost sound like impossible. So I don't know why, what contributed to their success, but it's certainly a direction to explore. Yes.
So for this web tool, how did you decide on a set of descriptors to generate these models?
Right. So we use the open source software to use the fingerprints, extended fingerprints, ECFP. So we only use those descriptors. But you can, there are many ways to compute the descriptors, right?
Usually, how many features does the data have on the deep learning? One of the criticisms is the interpretability of the model, right? Is there a need for variable importance, or any thoughts on that? I mean, this domain doesn't need the variable importance or some kind of
Well, I guess at this moment, people focus on the performance rather than interpretation of the model. And also, we use about 1,000 features. We always there's something called hashing. You can compute many ECFP, but then you use hash to control the number of features you use. Yes.
How about the compute hours? How do they compare between these methods?
Right. Yeah. So we compared using MATLAB version of deep learning. That takes days. Then we converted to the GPU-based cafe. That's usually cut the training hour to hours. So and then we use multiple GPUs so that you can run thousands of jobs.
And what about like the support vector machine, Naïve Bayes?
Naïve Bayes usually run much faster. Support vector machine depends on the kernel. I forgot exactly and we only have the largest data set we have a couple of thousand, so that's manageable by support vector machine. But if you have millions of compounds, then support vector machine will be very, very slow. Yes.
So I can see what very different data sets. Can you elaborate a little bit more what are the neural network you use, like the recurrent network, causal network,
This is just a deep multi-layer deep neural network. Yeah.
Any other questions? Let's thank the speaker.
Thank you.